Hi everyone, today we're talking about the next step in trying to figure out whether or not a donor tissue will be accepted by a recipient's body. And we're going to focus on these little buggers right here on the surface of majority of the cells in your body. These are called HLAs, human leukocyte antigens. We know that an antigen is a marker on the surface of a cell. We call them human leukocyte antigens because they're primarily found on leukocytes, aka white blood cells, but they're really found on pretty much every cell in your body in some combination. And the HLAs are responsible for stimulating the immune response in your body, meaning that if I have a cell that comes into my body that my immune system doesn't recognize the HLAs for, well, of course, it's going to attack it because it's going to attack any antigen that it doesn't recognize. So if there's a mismatch, it recognizes as foreign. But if there's a match, then that means that this cell will be accepted into the body as a same cell as recipient. So our donor tissue has to match in HLAs to the recipient as well, not just blood types, but these HLA markers as well. Now, testing that determines whether or not these HLAs are the same in the donor and the recipient is called tissue typing, which makes sense because we're literally transporting one tissue person to another. We're going to be testing the similarity between these antigens. They're not going to be perfect. It's not going to be exact because there's lots of complexity to these HLA antigens, as we'll see in the genetics in a second here. But the closer that we can get to these HLA markers being equal to the recipient, the more likely the recipient is going to accept and not reject the transplant. So this has to be done before any transplantation of any tissue. We're going to focus on kidney transplantation, so that's what our patient is dealing with, but any transplantation has to have this tissue typing test. Now, HLA expression is going to be controlled by a set of genes in one specific spot on chromosome 6. You have, of course, two chromosomes 6, one from mom and one from dad. You inherit half of your MLA or HLA genes from mom and half of them from your dad, and they're all going to be in this spot right here on the chromosome that we call the major histocompatibility complex. Histo meaning tissue, compatibility means whether or not it's compatible or not. Makes sense. There are two main classes of these HLA antigens, and each of those classes has 3 different genes in it. So therefore, there are actually 6 genes inside the major histocompatibility complex, which makes this further complicating our process here. And of course, you have one of these from mom, a haplotype from mom, and a haplotype from dad, which means one haplotype will give you 6 genes, and another haplotype give you another 6 genes that may be the same or may not. That means that your leukocytes might have as many as 12 different HLA antigens with a variety of different attachment styles for antibodies to recognize. So this becomes very, very complicated. And guess what? When we're looking at HLA typing for kidney transplants, we'll only pay attention to HLA A, B, and DR, but each of these 3 different genes have hundreds of alleles difference. It's not whether or not I have one shape and I don't have that shape, like the majority of the genetics that we've done is either this or that, a capital A or a lowercase a. It's more like there's a triangle, a semicircle, a square, and all these different shapes are determined by the gene at one specific loci or locus, and there are 3 locuses that we're paying attention to on each of the 2 chromosomes that are in question. So there are hundreds and hundreds of possibilities. You can see how the coding for these genes gets very, very complicated. We'll settle in for just the allele group and not necessarily the specific HLA proteins and expression levels and such, but just know that we're simplifying a very complicated polymorphic gene expression group. Now, the large number of possible variations and combinations in these HLA antigens making it very, very difficult to find a match in the general public. So we're going to probably stick to family as much as we can. And it's pretty easy to see why this might be if we just take a look at some simple genetics here. If I have a haplotype from each parent and I have a hypothetical family of 4, yes, each of them could have a 25% chance of a different version here. However, they have a 50-50 shot from mom to get either a green or a purple one, and a 50-50 shot of getting a orange one or a blue one. So there's a much more likely chance that your siblings have a similar haplotype combination. In fact, a 25% chance that they'll have an identical one based on this 6 antigen match, because there's only 2 chromosomes. We can't mix up the chromosomes here whatsoever. So typically we're going to look for siblings in this case. Now, HLA typing has traditionally been done with serological techniques. We take the serum of each individual. Specifically, we're going to be looking at the antigens found in the recipient and the antibodies. No, excuse me, the antibodies in the recipient and the antigens in the donor tissue. And we'll put them into an ELISA test, just like you've seen way back when we were identifying antibodies attacking antigens in our meningitis tests. We still would have to have a secondary antibody with an enzyme that reacts to a substrate if it is attached to the primary antibody. That would be the HLA antibody. So if that HLA antibody finds the antigen, then the secondary antibody will attach to it and the substrate will react, and therefore we'll have a color change. That is well and good, but there's so many different combinations that it tends to be a very tedious test that way. So instead, HLA typing is now predominantly done using molecular biology, the gene expression with sequencing instead. We isolate the patient's DNA, we PCR the sites that are specific to HLA genes, and we sequence the genes to identify the HLA compounds. It's expensive, but hey, if you need an organ, you kind of will take whatever you can get and pay the price for the HLA typing. Once the donor-recipient test has been tested for tissue compatibility of the HLA antigens here, we're also going to conduct an antibody screening. So we're going to take a small amount of the organ recipient's serum, the blood that they have, and we're going to mix it with 60 different individuals. These are going to be mostly randomized individuals that have very specific HLA antigens inside them. The scientists can then determine how many different HLA antibodies a patient has in their blood based on how many of these tests produce a reaction. So if I'm looking for a kidney and I need a donation, then they're going to test my blood to see how reactive my antibodies are, regardless on whether or not the donor has a very specific sequence of antigens in their tissue. We don't want the possibility of any reactions taking place with the blood that might be associated with the tissue. So we're going to test the antibodies on a bunch of different individuals. Now, if we have all those cells taking place here, and my antibodies attack 30 out of 60 of those, then my antibodies are 50% reactive. And we're trying to get that number to be as low as possible. So a person that has, say, an 80% reactive antibody test might suggest that their organ recipients, their receiving of the organs, wouldn't go so well. Their antibodies are super reactive to all sorts of antigens. After the tissue typing and antibody screenings are complete, then we complete one last cross-match test. This cross-match test is going to take a small amount of the potential donor's white blood cells, and it's going to mix them with my serum. Again, we're looking at the antibodies in the serum to see if they're going to attack cells that come into my body. If we have a positive test where my antibodies, the donor antibodies, the recipient antibodies, attack the donor white blood cells, then that's going to indicate that a transplant cannot be completed with those cells because that recipient's body will reject the implanted cells, whether they're white blood cells or the kidney itself. My antibodies would just go and find the antigens that this particular donor has supplied. Now, if it's a negative cross match, then that means that no reaction has happened between the donor and the recipient samples. My antibodies have not gone and found and destroyed the HLA antibodies or antigens on the white blood cells, and that means that a transplant can be performed. So we are making sure we can find negative cross matches between the HLA antibodies and the HLA antigens and the antibodies in the recipient's cells. We're trying to make sure that the immune system of the recipient will not attack the HLAs of the donor. That's all there is to tissue typing. It's severely complicated based on genetics because antigens come in all shapes and sizes, but we're trying to get as close as possible to matches as we can. In fact, in this one here, we can see that the positive cross-match had 20% of the cells lysed. That means that 80% of them did not lyse, which is on the good side. We don't have necessarily too bad of a positive cross-match. We want no cell lysis, obviously. But as little as we can take and then suppress the immune system of the donor or the recipient, that would be a pretty ideal situation here when it comes to organ donation, since we have so few organs and so many more recipients that are needed.